back here. So I say the original paper on fluorescence correlation spectroscopy is actually about a chemical reaction. And what Doug Magde, Elliot Elsion, and Watt Webb were studying were the reaction of ethidium bromide binding to the DNA. And that is a paper that has a very, very large number of citations. And is in this paper, what is interesting is that all the theory is derived by changes in concentration which affect the chemical reaction. And that paper was using differential equation describing the system as a continuum. What I say from the very beginning is actually the chemistry of the system is made of single molecule, and this better we focus directly on single molecule, which are different than using differential equation describing the system as a continuum. So the idea to use particle interpretation of the fluctuation experiment appeared originally in the work of Rudolf Rigler and Manfred Eigen in 1974. And they always thought in this term of using a single particle. It never occurred to them that they have to use concentration. But I will add something which is very important, which is the observation of fluctuation from the same molecule. So all the molecules are the same, are equal one to the other, but we talk about the fluctuation from the same molecule. And the first demonstration of that is a paper by Zino Foldes-Papp, which appears in 2001 or almost more than 20 years ago. And he was doing the experiment in a very strange way, strange for me at least, because he was trying to dilute an experiment and showing that the correlation function was always the same. And, but that was not really a demonstration, and people discussed that he's not doing the right thing. But we use a totally different approach, which is the use of spatial correlation for FCS. And that was developed a few years ago, and you heard a lecture about the Riggs principle. We jump other techniques that use spatial correlation, but essentially the first experiment on the Riggs were published in 2005, and then the pair correlation appears in 2008. So what is that? So I said that FCS and spatial correlation was used by myself and Dr. Dickman in 2005. And the spatial correlation have the potential to provide a more complete description of transport of single molecule in non-homogeneous system. In fact, if you look at the lecture that Nick was given, he shows a Gaussian and says the Gaussian will broaden, and as it broadens the Gaussian, because the particle can be found at different time in different position out of the original position, he used a Gaussian for that. And the equations that we're using for the in the Riggs are Gaussian. But this cannot be true. Everybody working with Excel knows that there are membranes, there are nucleus, there are organelles, there are many other things. So a molecule will not move according to a Gaussian, but will eventually hit an obstacle. And so the question that I posed in that work was, can we figure it out where the obstacles are and the position of the obstacle and the distance of the obstacle? And so we will change the language that we use, because we're not measuring diffusion, but we're measuring the presence of obstacle. And actually, we're measuring the diffusion of molecule in the presence of obstacle, which is something that, as far as I know, has not been done. Okay, so let us see where is the point. So the point is that if we measure an observation volume, which is drawn here, and molecules are moving in and out, so we measure the fluctuation, and we have now the correlation function with -1 because we are not using the fluctuation, but we're using the fluorescence. And that was done originally. This correlation function was written by many people working light scattering and in other things, but it's used normally in FCS. And one important change was done by Petrashvili and Jonas Rice, in which they use two foci. So using 2 points of observation, which are separated, they can tell if the fluctuation occurring in one point will propagate to the fluctuation occurring in the other point. And this goes under the name of dual foci FCS, because there are two volumes of observation, and that is done by measuring one part and the other. So now the question is, this is done by 2, which is interesting, but what if we have 3 and 4 and 5 and 6 and 7 and have many volumes of observation? So let us see what is happening. So the pair correlation concept comes from if we can perform a measurement of single molecule, but in a sea of many other molecules, which are identical. So this is it looks like very difficult and impossible, but let us see what are the concepts. So our volume of observation is like a camera in which we can measure the fluctuation in 0.1, 0.2, 0.3, 0.4, etc., 5, 6, 7, you can label those. But suppose that we have, for example, a membrane, which is indicated by this line in blue. So a fluctuation which is happening in 1, or a molecule which is at 1, can be found in position 2. But a molecule which is moving in position 2 cannot find in position 3, because it has to pass through a barrier which is impenetrable in this model. And a molecule which fluctuates in 3 can be found in 4, and so on, or can be found in the side. So this is the way which we introduce the topology of the space in order to tell when the molecule which is in 1 can be seen in 2, but if it's seen in 2, cannot be seen in 3, because the blue indicates a barrier. So suppose we have an obstacle. So, and we have an obstacle, so we are moving a particle, for example, this particle is moving here, but cannot cross the obstacle, but eventually can go on the other side if it finds the end of the obstacle, and then can be found in this space. But the path that this molecule is following, so all this path, is very different than the direct path. So if we measure the time it will take a molecule to go from one point to another, we can tell if it went to a long path, which is always a possibility, or it can go through a shorter path. So we will measure the time it will take a molecule from a given position to go in another way. So in this case, there will never be correlation between 2 and 3, because it's a barrier. But here we can go from this position in the other if there is a way to go around the barrier. So if we correlate in time the fluctuation at the many point in the grid and measure the time for a particle at a given point to appear at another point, we can have an idea of how is made the space around the particle. And this is the purpose of this talk. So let us now try to go to the mathematics. And the mathematics is actually, so mathematics we already did, but suppose we have a particle which is in position 0, and this can be indicate the position 0, 1, 2, 3 is a line. So if a particle is in position 2, as a function of time, we can find the particle in a parabola, because it goes with the distance traveled with the square root of time. So if a particle is at position 1, it can be found in another position, which can differ from, but you have to wait some time for the particle to go up to that position. So if we consider a point which is very close to the original point, and we do the product of the intensity at this point with the intensity at this point delayed, so this is not a correlation function, but it's the product of 2 intensity, we can find that if the 2 points are very close, the intensity, so the product of those 2 probabilities will be very large. But as the intensity, as we go farther and farther, so we increase the length, the time in which the particle can reach the given value, well, at the very beginning will be 0, because if the particle is here, cannot be there. So, but as the time goes on, eventually the particle can be seen. And so that means that originally the particle, the product of this intensity by intensity in another point will be 0. But at some point, the particle can be seen and then cannot be seen, or the correlation function will go to 0. But this is not correlation function. This is just intensity. Okay, so let's go on and see what happens about this. So the pair correlation, let us do first of all the one-dimensional thing. So the particle can be one point, another point, another point, another point, and another point. And I will draw that as determine the position of the particle as a function of time. So this is not a normal space, because in this coordinate we have a distance from the origin, and in the other case we have time. So this is a strange space, but still we can do the correlation function and calculate the correlation function and see what happens. Okay, so let us take 2 points at a given distance and let us see what happens. So a fluctuation which is in a given point, for example, this is a particle crossing that point, will appear at a later point at the distance r, will not be simultaneously seen at the distance r. So essentially, all the correlation are calculated in the following way. So I want to calculate the correlation function between a particle with a certain time delay and a certain difference in position, which I indicated with the r. And now I use the function, still the fluorescence, so I subtract 1, and I normalize in the usual way, exactly like we did for the correlation function. Okay, so essentially what I am trying to say is that the particle, so the delta t at a given distance will depend on the distance, and the delta r will depend also on the delta r, because of course, if I go very far, it will take much longer to get to that. So now this is the real point of the method. So I will say that only the same particle will produce an average positive cross correlation within a given time delay at 2 different points. And why is that? So this is the difficult part of the thing. So suppose those are the 2 volumes. So one will be here and one will be at a different position. Now, clearly, I have one particle passing through that observation volume, fine, and I will have another particle, different particle passing in this other, but there is no time relationship between what will happen with the green particle here and the black particle there. So they can be that the correlation is at time 0 or a very long time, because there are 2 different particles. Okay, and I subtract that by subtracting that one event. Okay, now let us see what happens if I go ahead with that. And so the same particle will never, okay, so the same particle will never correlate with itself, because if it's in this position, it cannot be in the other position. But if we go on with the same reasoning, okay, and now we consider one particle that was there, and the same particle crosses the other volume, it will have a positive correlation instead of 0. And that another particle will also have positive correlation. So I will have positive correlation only if the same particle can be, that was in one original volume, or can be found in the other original volume, of course, with some delay. And the delay will depend on the particle path and the particle is following. So first of all, let us do some simulation, because simulations are always easy. And I have only one particle. So only one particle will change the position like that, and I can calculate the correlation function between that point and another point. And of course, I will find a correlation function, and I call that correlation function of 1, which indicate that the distance is 1 pixel. 2 means 2 pixels, 3 pixels, 4 pixels, and so on. And you can see that the pair correlation function indicate if there is a cross correlation at the distance of pixel. Of course, a pixel, if I have a distance of 1, will always correlate. But if I have, as I shown here, farther and farther distance, it will not cross correlate. Actually, if you pay attention, the PC of 6, for example, in this simulation, goes slightly below 0. This is important. Okay, so So this is, so when the distance between 2 points is small, like in this case, points are within the same point function will produce correlation of the intensity fluctuation at a very short time. But as the distance increases, so it go to 2, 3, 4, 5, 6, the time correlation could start at very low amplitude because if it's in one position, cannot be in the other, but not only at a very small particle, but eventually can be negative. So this is very different than the dilution experiment of Xenofolder's part, because here we measure a cross correlation directly between one particle and another particle at a certain distance, while he was measuring the autocorrelation function at the same position. So that means the following thing, that if the distance is very small and or the diffusion is very fast, the cross correlation will have a maximum at very short time. As the diffusion becomes shorter, well, or longer and longer, the point of the maximum will shift. And so we'll go from green to red, eventually to blue, when it's very small. So essentially, the cross correlation of the two particles at a given distance will have a maximum, not close to zero, but a certain distance, because it has to travel that a certain distance in order to be able to see. And now you can see something that if in a point we have two kinds of particles that move slow and move fast, what happens is that we will have like first a correlation of the particle that move slow, and then a correlation of the particle that move fast. Move fast and then move slow. So this is very, very important because this is a phenomenon that everybody knows. So if you diffuse particles in a medium which contains obstacles and so on, you can have different apparent times of presentation of the particle depending how if you diffuse faster or diffuse smaller. Notice also that in this scale, essentially the width of the correlation function does not change very much. And this is also important. And if we go very small particle, the probability start, or the pair correlation function start below zero. And this is important because that means if you start below zero, that is the particle is in one position, cannot be in another position. Simply, this is the only thing that we say. Okay, but let us see what happens in one dimension, and then I will show the two dimensions. So originally, the particle is here, the particle can diffuse everywhere. So if we can make a circle around the particle, so if we make a circle and the particle can only move in a plane, always I will find the particle with some delay, but always I will find the particle. The probability to find the particle depends on how big is the volume of observation. So it will depend on the length of the circumference, which is equal to the pixel position, and the point function, which can be anything I want, essentially. So this is what is happening. And so later, everything up until here is reasoning and nothing else. But now let us do an experiment. So we have bid in solution. So if we have bid in solution, so those are the bids, and now I have here the position along a line, and here the time. So you can see that if I do the autocorrelation function, as it says here, I found some correlation at a very short time, and eventually that will decrease. But if I do the pair correlation function, I found a very different pattern. So I found no correlation at very short time, but a large correlation at a given delay when the particle will be found in the other place, and then eventually will go to zero. So you can see that this pattern is very different than the other pattern, and it depends on the fact that the particle can be moved from one position to another. But it's the same particle. It cannot be anything that more than one particle. And the way to show that is simply to have only one particle in the simulation. If I have only one particle, well, it has to be the same because there is only one. And that pattern is independent of the number of particles. Is simply the delay gives me the time it takes a particle to go from one point to another in time. But notice that all the bits have the same delays because this is an experimental value now. It's not a simulation anymore. So now let us see some consequence of that. So some consequence, which started with the work of Liz Inde when she was a postdoc in my lab. So suppose the particle can be in the volume D1, and suppose this is a border, for example, of chromatin, and then a particle can be in D1, in D2. So if I do an experiment and I put the point here and the point there, this is an impenetrable barrier. Suppose it is an impenetrable barrier, so the particle can never be found there. It can never be found, so with respect to the center of the particle, in the border left and in the border right, I will have no possible communication because the particle cannot enter that. But if once in a while the chromatin opens up and the particle can enter, I can find something. And you can see that finding will end up that I have a delayed communication, which will depend when the chromatin opens up and the particle can go there. So that means that if I have no communication, I will have a black line. If I have communication, but delayed communication, I will have a circle or a semicircle. And this is so easy to see in the data and so simple to see that I just have to look for impenetrable lines or lines which communicate at a given distance, which will always be that sort of semicircle. Well, it's not a circle, but has that shape. Okay, so let us see what experiments say. And in order to do this experiment, I will study a particular system, which is a replication of nucleoplasmic shuttling. So if that is the cytoplasm, we have molecules in the cytoplasm, and the molecule can pass through the nuclear pore complex. If the molecule is small enough, it will pass directly to the nuclear pore complex by diffusion, which takes a lot of time, and will get into the nucleus. If the particle is large, it needs to have a active transport, so it means to have we have to give energy. And what happens is that we have molecules in this side which will bind to the particle to be transported, will interact with molecules in this side, in the nucleoplastic, in the cytoplasmic side, and eventually will be transported in the other side. So let us see if we can distinguish between passive diffusion, which are small molecules just diffusing to the nucleus. And this will be bidirectional because it can go in one side and the other. And this mechanism is known to be regulated by molecular size. And for molecules larger than 60 or 70 kilodalton, they cannot pass through the nuclear pore complex. And this energy independent diffusion. But if we have active transport, so in active transport, there is unidirectional through the nuclear pore complex. So molecules which are here can be found in the other one. And those are driven by specific amino acid sequence, one for nuclear localization signal, so a molecule which is here can be found in the other side, or nuclear export signal, so a molecule which is here can be found on the other side. And this is energy dependent. So here is shown what the reaction is. So we have a particle, for example, a GFP, which has a nuclear localization signal, will interact with importin alpha, and then importin alpha will interact with importin beta, make at least a 3 protein complex, and that complex will pass through the nuclear pore complex. And so this is the complex in the way, well, it's my drawing, and then the complex will be found in the other way. Of course, that will be totally independent of the direction, but if I can dissociate this complex in the nucleus, then it cannot go back in the same way. And that is really how nature works. So nature works with a protein, which is a RAN GTP, which interact with importin beta and eventually liberate the protein that needs to pass through the nuclear pore complex. Of course, the concentration of NGDP has to be regulated because you need energy for this process to occur. Okay, and so the NGDP is known to have a very high concentration in one part of in the nucleus and very low concentration in the rest of in the cytoplasm. So in the cytoplasm, you can form the complex. It will not dissociate because the concentration of NGDP is very low, but as soon as it passes through the nucleus, it will dissociate and the particle will be found in the other part and cannot be returned. So, and this is due to the fact of the interaction that the protein will have with the, well, first of all, it needs to have a signal, nuclear localization signal, and then at the end, you find that protein in the other side, of course, at the expenses of the energy of NGDP. Okay, so first of all, people have studied this phenomenon for many years, and they have found that for 10, 27, or 50, up to 57 kilodalton protein, passive diffusion is lowered by 100-fold at the nuclear envelope boundary compared to diffusion within the nucleus of the proton, as expected. So this is very slow because the particle has to diffuse through the nuclear pore complex. And that kind of experiment provides ensemble averaging measurement. Passive and active flux cannot be discriminated except by the time. But the single molecule experiment were done and were shown that actually molecules in only 10 milliseconds can pass from the one part to another part of the nuclear pore complex. So let us see if we do pair correlation function of this experiment, what happens. So this is single molecule experiment. You see, this is the cytoplasm, this is the nucleus. So the molecule, the complex common stop there, and then after some time disappear, and you can eventually, if you're able to track the particle, you can find in the other side. Okay, so let us see if we can do that sort of experiment in the following way. So this is a nuclear part, this is the cytoplasm part, and the protein has a nuclear localization signal, which makes the concentration in the nucleus much larger than in the cytoplasm. So we draw a line through that point. So a line to that point means that we have points which are in the nucleus, points which are in the cytoplasm, and a point which are, for example, that point, and that point it will be one in the nucleus and one cytoplasm. So we have all the conditions that we need to apply the pair correlation function. So first of all, the pixel size is about 100 nanometers. So, and we have a total of 32 points in a line. This is important because it will give us a chance to observe several points in one side, several points in the other, and the boundary. What happens at the boundary? So if we do, so the point spread function is on the order of 200 nanometers, so it's about 2 points. And we correlate all the points which are in that side, and then we can continue to correlate with points which are in the other side. But let us see what we found. So it is important here that we have the time. So the time of each pixel, pixel 1, pixel 2, is 6.3 microseconds. The time of a different lines, so one line to respect to the other, is 0.74 milliseconds. So we are in the range that we can measure passive and non-passive and active transport. And then we can measure correlation between points in the nucleus and correlation within points which are in the other side. And let us see what we found. So first of all, this is the nucleus, which is very bright, and this is the cytoplasm, and this is the nuclear envelope. So if we do a simple ACF analysis, we see that here will be larger because there are less molecules, and here will be smaller because they are less, but it's a continuous function essentially. And if we measure that, if we measure the time it takes from red to blue, we can find that, for example, column 3, in column 3, the time is 11 nanoseconds, but to get in column 28, which is from here to here, it takes much longer, as you can see from the correlation function. Okay, so now let us show the 32 correlation functions that we have. And we see something very interesting. So that everything which is in the cytoplasm move or in one side of the thing moves very fast, move very slow, but as soon as we get in the nuclear envelope, we have two correlation functions that are completely different than the other. And those two correlation functions are exactly at the border between the nuclear envelope. And so there are two events, one with a diffusion constant, which is 8.1, which means we're moving, for example, in the nucleus or in the cytoplasm, and then one which is the crossing of the part, which occurs with a totally different time. So essentially, if we do the correlation in the nucleus, the correlation in the cytoplasm, so nucleus to nucleus, and then here we have the nuclear envelope and the cytoplasm, we found very time. And actually, not only that, but you can see that at some point, for example, at a given distance, we found the black line, so which means that there is no communication between one part of the other. There is no communication at this time. So that will be the autocorrelation function. That will be in the nucleus or in the cytoplasm. Then if we increase the distance, we can see that as we increase the distance, we get a bump in the curve, which means that the particle arrives after a certain time. And if we do all this time, and it appears that the nuclear envelope is crossed with essentially a millisecond. This is, I don't know if you can see that. So those are seconds. So in about a millisecond it crosses, which corresponds to what was measured by other people. So, so first of all, and this is a large distribution, as it should be because it's diffusion. Okay, but now, so the passive diffusion is low, as we can see there. But if we look at the cross of the nuclear to the cytoplasm, actually, we found a very different correlation function and much faster. So which means that the correlation function due to shuttling is much faster than the other one. And here is the evidence of that. And you can see that this is not anymore 150 milliseconds, but it's about 3-4 milliseconds, as you can see from this part. And notice that initially it's negative because if the particle is in one side, cannot be in the other side. And that proves that we are measuring the same particle. So we can measure directly the nuclear diffusion, the passive diffusion, and the active transport, as you can see from here. So, and you can measure many other things. For example, what is the input in alpha, what is the input in beta, and so on. And then let us do a control experiment just to be sure. So if the localization has to be ATP requires energy. So if we deplete ATP from a condition in which we have a lot of transport, so a lot of proteins found in the nucleus, from a condition in which have very little ATP, very few of the molecules pass through. So this is very important as a control. Then, and that gives us information about the transport. So let us go now to two-dimensional experiment. So in the two-dimensional experiment, we first of all, we consider V in cell 1 and the cytoplasm cell 2. Well, in cell 1 and cell 2 should not be crossing because they have to jump between one cell and the other. Well, some molecules do that. So what we have is in intensity, we have a gap very clearly. But in correlation function, we have no communication, so it's a black line. And that proves that essentially molecules cannot jump from one side to the other. And if we measure that, we found a very, very long correlation function eventually. Something will pass through, but it takes a lot of time. Okay, now let's go to the second part of the talk, which is about the two-dimensional. So first of all, the pair correlation function has that particular shape. So now we measure at one angle and another angle and another angle and another angle. We measure all the possible angles around the particle. And the correlation function changes. So that means that in one direction is more probable, so to pass than in the other. So that means that if we are far from a barrier and we measure the correlation function in 32 directions, for example, it will be around, essentially all the directions are compatible. But if we have a barrier in one side, so what happens is something very different, that the particle cannot go in this direction because they will hit the barrier, but they can go in the other direction. So you can see that the barrier determines the shape of the pair correlation function as a function of the angle. Okay, so that means that we can determine where the barrier is just by looking at the shape. And this is, for example, a simulation in which we have a region where we have molecule, another region we have a different concentration of molecule. And what we observe is that one barrier here and one barrier there. So every time we have molecules have to go in this direction or in the other direction, we found essentially that there are barrier in one side and barrier in the other. So one single barrier gives two correlation function, which will depend on, so the angle of the value will change in this particular side. So if we have a channel will have three, 1, 2, and 3, because there is barrier here, there, and barrier there. And this is what we found. And so the correlation function in one side will be deformed in that part. If we are in the center, will be deformed in both parts. If we are in the other side, will be deformed in the other side. So we can see in every case where the deformation occurs. The deformation gives us directly the position of the particle. And this is different kind of for example, a circular particle, we have only two. And then if you have square particles, also we have two in one side and the other. And finally, if we have a cross or any other thing, we can see the particles going in one direction, but not going in the other direction. So we can tell where is the barrier, what is the shape of the barrier, at least locally, and if the particle can cross or not. And this is to finish a system in which we have many different parts in which the molecules can move here, but cannot jump to the yellow part. And then what we say is that we have the direction is uniform in this part, but there is another flow, which is in the yellow part, which are all communicating and communicate in the other direction. And you can see that you can determine the shape of the obstacle very well by this simple algorithm. Okay, so I think the same thing can be done in cell and so on. So let me jump to the summary. So the PC pair correlation function makes to measure molecular flow at essentially 100 nanometer scale. Well, I forgot to say that those are 100 nanometer, essentially. So you can determine that very well. Experiment will reproduce the prediction of the pair correlation function. Simulation show that we should be able to detect diffusion boundaries that are, for example, 200 nanometer apart. Experiment with molecules in cell show obvious barriers to diffusion at the junction between cells. Experiment with NLS EGFP show passive and active transport, and diffusion of the nucleus shows disconnected region of flow, for example, and so on. So I think this is the summary of this lecture. And so let us say that you can very well see single molecule. You can see molecules going from one part to another in the cell, and you can see regions in which common motions are communicating one with the other, and regions which are not communicating one with the other, exactly as the prediction of the basic equation will show. And I will stop here and open for questions.